Good morning, America. This is Nate Niemer along with Nico Piperis with another episode of Biology 101. Let me send it over to Nico.
For this episode, we will be diving in depth into two very important cell organelles, the endoplasmic reticulum and the ribosome. Ribosomes and the ER are closely related in their functions in the cell. They create proteins and other materials needed by organelles to function properly. Specifically, ribosomes are even attached to rough ER for direct interaction in creating these proteins.
First, we'll look at ribosomes. Ribosomes are composed of two protein subunits and a ribosomal RNA. The ribosomal RNA carries out the basic production of the proteins, while the subunits act as a catalyst in their formation. The ribosomes are produced and assembled in the nucleolus to form one of the smallest organelles in the cell.
Although one of the smallest organelles, it has one of the most important functions in the cell. It is responsible for the synthesis of many different proteins. There are two types of ribosomes, free and bound. Free ribosomes float throughout the cytoplasm of the cell, while bound ribosomes are attached to either the ER or the nuclear envelope. Both of these types of ribosomes are identical to each other in their structure.
Free ribosomes, as mentioned earlier, float freely in the cytoplasm. These ribosomes create proteins that function within the cytosol. They catalyze the first steps in carbohydrate breakdown.
Bound ribosomes, which function with the ER, create proteins that insert themselves into membranes, are secreted outside the cell, and insert themselves into other organelles to help perform their function. For example, proteins located in the lysosomes are used to perform intracellular digestion. Without ribosomes, the cell would basically lose all of its function, considering that the proteins ribosomes make are responsible for digestion, transport, and cellular respiration.
Now, on to the endoplasmic reticulum. The endoplasmic reticulum, commonly referred to as the ER, is a network of membranous tubules and sacs called cisternae. The internal cavity, known as the ER lumen, is separated from the cytosol by the ER membrane. The ER is made up of 60% of the membrane found in the cell. The ER and Golgi apparatus interact frequently. The different materials created in both the rough and smooth ER are transported to the Golgi apparatus by transport vesicles, which originate in the ER. The Golgi body then modifies the products to their final conformation and ships them to various locations in the cell.
There are two types of ER, both of which are located directly next to the nucleus. The smooth ER, which lacks ribosomes, performs four important cell functions. It creates lipids, metabolizes carbohydrates, stores calcium ions, and detoxifies poisons. Because of their specialized function, different cells have various quantities of smooth ER. For example, the liver, which detoxifies most poisons in the body, contains a much greater amount of smooth ER than an average human cell. If the smooth ER was damaged in some way, the cell could not break down dangerous toxins or create the lipoproteins that carry various lipids throughout the body.
The rough ER is directly connected to the nucleus through the nuclear envelope. The rough ER's only structural difference from the smooth is the presence of ribosomes on the outside of the membrane. This slight difference is responsible for a dramatic change in function between the two. Rough ER's main function is the synthesis of secretory proteins called glycoproteins. These proteins are bound for various parts inside and out of the cell. These proteins are contained in transport vesicles that move the proteins to the Golgi body and other organelles, where they will perform their main function. Along with protein production, rough ER also synthesizes membranes in the cell. Without the rough ER, the certain organelles would lack the necessary proteins needed to perform their function, such as the mitochondria and lysosomes.
Thank you for tuning into this episode of Biology 101. We would like to thank our main sponsor, the Cleveland Browns, and wish them the best of luck in their upcoming season. We hope that you'll tune in next week for our civil discussion on neurofibromatosis. For Nate Niemer, this is Nico Piperis, reminding you to stay classy and go Browns.